Thank you for the kind introduction. Today, I will talk about the multidimensional evaluation of ALS mouse models using preclinical 7 Tesla MRI as shown in the photo here. I have no financial relationship to disclose. So, bridging ideas from basic research to clinical application requires overcoming significant hurdles. The importance of reverse translational research from the clinical field is emphasized in addition to translational research from basic research. MRI provides a common assessment method between animal and humans, effectively bridging this gap. In drug development research, MRI can make significant contribution to pharmacological studies. In addition, MRI can be used for pharmacokinetic studies and human clinical trials. Today, I would like to bridge the gap between preclinical and clinical studies by introducing pharmacological and pharmacokinetic studies within preclinical research using O7T labeled water. The main topics of today's symposium is O7T MRI, which we evaluate using the indirect method. Gadolinium and iron contrast agent affect both T1 and T2. This leads to a complicated concentration-dependent behavior of the MRI signal. While low concentration of these agents mainly enhance the signal due to T1 shortening, higher concentration lead to signal decrease due to T2 shortening. In contrast, O7T only affects T2. The lower figure shows T1 maps and T2 map obtained by imaging phantoms with different O7T concentration. T1 map shows no concentration-dependent change, while T2 shows concentration-dependent signal changes. And Dr. Harada of Hokkaido University reported in 2022 that O7T concentration can be easily estimated from the MR signals. Taking advantage of the characteristics of O7T MRI, we began to study ALS. ALS is known as a neurodegenerative disease that affects motor neurons. In Japan, it takes an average of 13 months from the onset of ALS to hospital diagnosis. In April last year, the FDA approved a new drug to inhibit the progression of ALS. This has increased the importance of early diagnosis. We therefore investigated the potential of using O7T MRI for early diagnosis of ALS. Early impairment of the blood-brain spinal cord barrier and swelling of perivascular astrocytes are known in human model of ALS. Gadolinium contrast agents cannot evaluate this barrier leakage sufficiently. Therefore, we decided to evaluate abnormal brain water dynamics using O7T labeled water. We focused on SOD1 mice, which are ALS model mice expressing mutant human SOD1. First, because paralysis usually began with the hind limb impairment, behavioral abnormalities were identified by the assessing hind limb scores. We assessed body weight, general condition, hind limb response, and the extent of spray during falls. As abnormalities were observed at 15 weeks, we defined this as onset. MRI was performed at 11 weeks when no abnormalities were observed, and at 60 weeks as post-onset. Two MRI studies were performed, one with PSO-17 administered intravenously and the other with high concentration O7T labeled water administered intracisternally. MRI can provide different contrast, allowing for multidimensional assessment. We measured T2 weighted image, dynamic O7T, gadolinium contrast scans, DTI, and T2 maps. First, we present visual observation from T2 weighted images. Mild hyperintensity of the trigeminal motor nucleus and facial nerve was observed on T2 weighted image after the onset of ALS. Hyperintensity on T2 weighted image has been reported in previous studies. In addition, we statistically compared voxel by voxel T2 weighted T2 maps and found significant difference in these regions. Next, we present the results of DTI analysis. Fractional anisotropy FA, indicating water diffusion anisotropy, decreased significantly after the onset of ALS. Similarly, decreased FA was observed in the trigeminal motor nucleus and facial nerve. Professor Abe of the University of Tokyo reported significant CST FA reduction in human patients in 2004, confirming our findings across species. We observed the dynamic MR signal change caused by the gadolinium contrast agent. Rapid injection was performed 3 minutes after the start of the MRI measurement, and the measurement continued for 15 minutes. The coordinates of the anatomical image of each individual were normalized, and the average map of each group and its difference image were generated. The AUC was determined for each of the post-injection AUC, and an increase in gadolinium contrast effect was observed in the cerebellum after the onset of ALS. Similarly, we observed water dynamics after the intravenous injection of PSO-70 using indirect methods. No significant difference were observed due to sensitivity issues. We then administered high concentration O7T labeled water intracisternally to observe water dynamics. We administered O7T 15 minutes after the start of the MRI acquisition. Measurement continued for 60 minutes after administration. In particular, abnormal O7T accumulation was observed in the cerebellum before the onset of ALS. Quantitative AUC comparison showed significant difference. The lower movie showed that in the cerebellum, the influx of O7T labeled water is greater in the surrounding CSF region and in the middle cerebellar peduncle region. Abnormalities were also observed in the CST after ALS onset. In summary, we defined ALS onset by behavioral analysis and performed MRI measurement before and after onset. Abnormalities were observed in the motor nucleus of the trigeminal nerve and the facial nucleus on T2 weighted image and FA after ALS onset. These nerve control facial muscle, indicating the observed functional decline after ALS onset. CST is already known to be abnormal in human patients, and abnormalities have been found on FA and O17 intracisternal administration. It is thought to reflect descending motor neuron dysfunction. And no abnormalities were observed in the primary motor cortex. Since this experiment was a comparison of pre- and post-onset ALS, we observed ALS in the early phase. It may be possible to detect abnormality in the motor cortex by targeting ALS in later stages. Abnormalities were detected in the cerebellum before and after the onset of ALS using O70 MRI, especially in the middle cerebral peduncles. These fibers, which originate in motor cortex, transmit motor information and represent descending motor pathway. MRI allowed multidimensional visualization of ALS-induced motor neuron dysfunction. In particular, O70 MRI uniquely detected abnormalities in the middle cerebral peduncle before the onset of ALS, potentially supporting early diagnosis. Based on these data, we are planning a clinical trial in ALS patients. So finally, let me to make an announcement. Our research institute will officially change its name to Jichuken from next April. In addition, at the end of last November, we installed 11.7 Tesla small animal MRI. From mice to marmoset, various imaging methods are available. If you have interesting models or experiments, please let's collaborate with us. Preliminary data show the ability to image the whole body of the marmoset with a resolution of 100 micrometers using new MRI. And this project was conducted in collaboration with Professor Kudo and Dr. Kameda of Hokkaido University with the support of many persons. I'd like to take this opportunity to thank you all for your cooperation. That's all. Thank you for your attention.